Hello students. In today's lecture, we are going to discuss flavonoid test performed in ethnobotany practical classes, and this comes under phytochemical tests in ethnobotany. So let us revise what are phytochemical tests and what are phytochemicals. So as we have discussed in previous lectures, that phytochemicals are plant secretory metabolites which play many roles including defense mechanism, responses to environmental stresses, and they have antioxidant and antimicrobial anti-inflammatory properties. And plants, phytochemicals can be extracted from different parts of the plant. Knowledge of these phytochemicals are important because they are utilized for the synthesis of drugs, medicines, cosmetic products, etc. And there are many phytochemicals which are present in plants which are synthesized as secondary metabolites, and they belong to alkaloids, steroids, tannins, glycosides, phenols, flavonoids, saponins, etc. So, in the previous lecture, we have already discussed what are alkaloids, what are saponins, and what are tannins. In today's lecture, we will be discussing about flavonoids. So, what are flavonoids? Flavonoids are secondary metabolites belonging to subgroup of polyphenols. They are responsible for giving color to flowers and fruits, vegetables. They exhibit vivid colors. So, besides giving color, flavor, and fragrance to plants, they are helpful in attracting pollinators. They are synthesized in plants in response to microbial infection. They have antioxidant activity, and some well known flavonoids include quercetin, camphorol, catechins found in food and fruits like citrus, berries, and tea. So far, over 10,000 flavonoid compounds have been isolated and identified, and they are used as natural dyes, cosmetic and skin products, as well as anti wrinkle agents. Most of the flavonoids are used as therapeutic agents, and they have been used as anti cancer, antimicrobial, and antiviral agents. Citrus fruits are a rich source of flavonoids. So, when they are used in chemotherapy, and when we have seen that these flavonoids are less expensive compared to the commercial drugs which are used in chemotherapy. So, what are the examples of flavonoids? So, some of the examples include quercetin, which is a flavonoid plant pigment present in plants such as onion, green tea, apple, berries, and red wine. And this is commonly consumed as it has antioxidant, anti-inflammatory effects, which helps in to reduce swelling, kill cancer cells, control blood sugar, and helps in to prevent heart diseases. Camphorol is another flavonoid which is natural, and it is found in tea, beans, broccoli, apples, and strawberries, as well as aloe vera, ginkgo biloba. Camphorol has anti-inflammatory and antioxidant activity. And another example of flavonoid is catechins, which belong to flavonols of family of flavonoids. They are mostly abundant dietary sources are green tea, cocoa products, and wine. So these are some of the examples of flavonoids which I have taken. Besides these examples, there are several others. So in today's lecture, we are discussing the phytochemical test for performing flavonoid test. And so, what is the principle behind this? The principle of this test is that it involves treating the sample with different reagents and observing the color changes. For this, we will be performing two tests which are called alkaline reagent test and lead acetate test. So, what are the requirements of this practical? We require fresh leaves. We can also do this practical with dried leaves, but we are doing this practical with fresh leaves. And what are the chemicals which are required? 10% NaOH, 10% lead acetate solution, any dilute acid, which can be hydrochloric acid or sulfuric acid. The glassware includes conical flask, beakers, test tubes, measuring cylinder, pipettes, pestle mortar, cotton, filter paper, funnel, stand, muslin cloth, distilled water, etc. So how do we do this practical? So the first step is extraction. During extraction, we crush 20 gram of leaves using a pestle and mortar and make a fine paste with 100 ml of water. So as we have already discussed, the ratio of leaves and water will be 1 is to 5, and this paste will be filtered through muslin cloth, and this filtrate will be used for tests. So for flavonoid test, as we have discussed, there are two tests: alkaline reagent test and lead acetate test. So in alkaline reagent test, to this 1 ml of extract, we add few drops of 10% NaOH in a test tube, and the formation of intense yellow colored uh, indicates the presence of flavonoid. When we add a few drops of dilute acid, this color disappears. And the second test is lead acetate test. So to 1 ml of extract, we add 10% of solution of lead acetate, and the formation of yellow precipitate indicates the presence of flavonoids. So what is the difference between alkaline reagent test and lead acetate test? In alkaline reagent test, we are getting intense yellow color, but in lead acetate test, we are getting yellow precipitate. So we will see the difference, what is the color and what is yellow precipitate in subsequent slides. So like here you can see that first picture is of Combretum indicum, which is Madhumalati plant, and you see alkaline reagent test, you can see the color, it is intense yellow, while in lead acetate test you can see that a precipitate has formed, and this is not intense yellow, this is light yellow. In the second picture, which is of Tradescantia specia, so you can see that in lead acetate test, the test is negative. There is no, although the precipitate has formed, but it is not yellow, it is green, so we will consider it negative. And there is positive in test in alkaline reagent test, because you can see intense yellow color and there is no precipitate. So the, although yellow color is there, but there is no precipitate. Now the third picture is of Carissa carandus, which is your Coronda plant. So you can see that in alkaline reagent test, there is dark brown color, and in lead acetate test you can see that although yellow color is there and the precipitate is formed. So for this Carissa carandus, we will consider this as negative. Alkaline reagent test as negative, you can consider positive because probably this is yellow, and then which is a dark yellow, and lead acetate test is positive. Now in citrus, you can see that lead acetate test and alkaline reagent test both are negative because there is no yellow color. Similarly, in Gymnema sylvestre, which is Madhu Nashini, you can see that alkaline reagent test and lead acetate test both are negative. When you compare all these plants, you can see that in the first picture, which indicates three plants, Combretum indicum, which is your Madhumalati, you can see that alkaline reagent test is positive, and lead acetate test is also positive. You can see that alkaline reagent test is showing yellow color while it is showing precipitate in lead acetate test. When you see Caronda, which is Carissa canandus, you can see a slight positive test, or you can mark it as negative also. And you can see in lead acetate test, the precipitate has formed, but it is not yellow, so we will consider it as negative. In Tradescantia, you can see that the positive test for alkaline reagent test, which is yellow color, and negative for lead acetate test. While in the second picture, you can see for asparagus, for citrus, and Azadirachta indica, all the tests, alkaline reagent test and lead acetate test, are negative because there is no yellow color or yellow precipitate. Although you can see that precipitate has formed, but it is not yellow. So we are considering negative test for asparagus, citrus, and Azadirachta indica. So these are some more pictures which we had performed earlier in earlier batches. So you can see that in Gymnema sylvestre, you can see that brownish yellow color has come, and in asparagus the test is negative. In Combretum indicum, you can see a slight positive test, and in Vinca rosea and Asparagus officinalis, you can see that the test is negative. So, this is a table where you compare the tests which are performed in different plants. So you can see that in row U, the test is positive for alkaline reagent and it is negative for lead acetate. In Madhumaliti, which is Combretum indicum, you can see positive test for alkaline reagent and negative and also positive for lead acetate. In Madhunashini, which is your Gymnema sylvestre, negative test for both tests. In lemon, which is your citrus lemon, negative test. Neem has also negative for both alkaline reagent and lead acetate test. Shatavari, which is asparagus, is negative, and Coronda, you can see positive for alkaline reagent and negative for lead acetate test. I hope the practical is clear to you, and let us meet in next lecture. Thank you, students.